For this video, I'm going to explain a treatment that involves the phages. And for this, the microorganism that I choose is a bacteria called carbapenem-resistant Acinetobacter baumanis. The original is Acinetobacter baumanis, but nowadays it has a resistance of a chemical that it's called carbapenem, and this microorganism is called crab. This one can cause human infections of the blood, in the urinary tract, and other body sites. The problem with this bacteria is that these bacteria are multi-drug resistant, and this thing make infections very difficult to treat. As I said, I'm going to use phage therapy. For the bacterial strains of the microorganism that I choose, that it's the Acinetobacter baumannii, I'm going to use a biotechnology technique that it's called pulse field health electrophoresis. This one is because I want to choose strains that ones are carbapenem resistant and the others are carbapenem sensitive. And with this, why I choose this pulse field health electrophoresis is because I want to analyze bacterial genetic differences between the carbapenem resistant and the carbapenem sensitive. As a second technique, I'm going to use multi-locus sequence typing. For this one, what I want to do is to describe the genetic backgrounds of the crab isolates. As I mentioned, my microorganism, it's called crab. The procedure of this method characterizes isolates of microbial species using the DNA sequence of internal fragments of multiple housekeeping genes. And the last method that I'm going to use is the multiplex PCR. This process amplifies the DNA sequence or the DNA in samples. Why I choose this method, it's because I want to detect some genes that are called oxacarbapenemase in the crab strains. As I mentioned, I'm going to use the phage therapy, so I will isolate and characterize a phage from the Acinetobacter baumannii, in this case in vitro, including its bacteriolytic activity and the whole genome. What the model that I'm going to use, it's a larva that it's called Galleria melonella, and this one, it has been used for several years as a tool to assess the virulence of bacterial pathogens. The phage therapy has many advantages and disadvantages nowadays. One advantage and one disadvantage that it has, the advantage is that the phages are very specific, so it cannot harm the useful bacteria that live in the body. And one disadvantage could be that are only few international recognized studies that prove the efficiency of the phages in humans. For this one, I select the phage therapy because it is an antimicrobial treatment with great potential for controlling infections such as the crab infections. Since their discovery, the phages have been used for therapeutic purposes and have shown their effectiveness both in vivo and in vitro without producing adverse effects or toxicity. And as the conclusion, I can say that the biotechnological advances have further expanded the repertoire of potential phage therapeutic to include novel strategies using bioengineered phages and purified phages lytic proteins. The current research on the use of phages and their lytic proteins, especially in against the multi-drug resistant bacterials, suggests the phage therapy, and there are the reference.